right, hey guys, welcome back. This is the last section for chapter 10. So chapter 10 we said was gene actions from DNA to protein. So in section 1 we talked about the types of RNA as well as what transcription is, how it's when we take a section of the DNA known as a gene and we make a copy of just that section in the form of RNA, specifically mRNA. And then we also had to process that mRNA before it could go to the next step, which was what we talked about in section 2, translation. So we would take the DNA and, we took the DNA, we use it to make the mRNA, we process that, and then the mRNA goes out into the cytoplasm where it gets picked up by a ribosome, and with the help of tRNA, it's able to build the amino acid chain, also known as a polypeptide. After that, we had part 3 of, I guess I should say step 3 of protein synthesis, which was protein folding. You take the polypeptide, you fold it up into a 3D shape, and it becomes a functional protein. Now that's just a simplistic view of it. We're not going to get into protein folding all that much. But now we are going to move on to section 3 of our chapter, and that is the human genome sequence reveals unexpected complexity. So now we're going to talk about how the genome is, well, a little more complex than what we like to pretend that it is. All right, so we got, all right. Sorry. So with our technology, we have been able to actually sequence genomes. There's a whole human genome project a while back. And since then, we've sequenced not just more and more people, but we've also sequenced more and more organisms. So other animals, plants, bacteria, viruses. By knowing their genome, it allows us to compare them to each other, just like we can compare human genomes to each other, find out where the differences lie. But it also allows us to look at and compare the genomes of different species. By knowing the genome of, let's say, a virus, we can understand a little bit more about the types of proteins it might need to make in order to take over a host cell, and thus we can use that information to help create vaccines, cures, medicines, etc. Now we have something called proteomics, and this is a study of the collection of proteins produced by a particular cell. So by collecting the genomes of a whole bunch of different organisms and sequencing them, like I said, we can find out more about the proteins that it needs. So once we analyze these genomes, we discover the proteins that they create, we can then study the types of proteins that are being made by a particular cell, whether that cell is a bacteria cell or a guard cell in plant stomata or in a human brain cell. We can even look at the proteins in one person, the different types of proteins that are made in a brain cell versus in a skin cell. So it allows us to do a lot of comparison research, and by knowing the proteins that are involved in these different types of cells, it does help us figure out more about how the cells work. It gives us a better picture of what's going on inside the cell and what it is doing as its primary job of keeping us as an organism alive and fully functioning. Genome economy, reconciling genes and the number of proteins. So most of our DNA is actually not encoding for proteins. We have a relatively small amount of genes. Only about 1.5% to 2% of our DNA includes genes. So those are protein encoding sequences. And yet somehow we're able to make over 22,000 different proteins within our body from such a small amount of DNA. And the question becomes, well, how does that occur? And a lot of it is because of different ways that we process the mRNA once it's been done. So once we've transcribed the mRNA, remember we talked about how we process it, we cut out the introns, and then we put the exons back together. And then from there, we now have a functional mRNA. But what if we process it a different way? If we process it a different way, that means then we have different bases located in the mRNA, which means we're going to have different amino acids, which means we can have a different protein. And that's what we're finding out, is that through processes such as differential intron splicing and exon shuffling, we're able to actually take the same original mRNA sequence, but make different proteins out of it. Now, how differential intron splicing works is, if you remember, we kind of said, let's say you take out these two sections, these two introns, cut them out, and then push the rest of it back together. Well, what if we left those two sections in and took out two different sections? So what we originally considered introns are now exons, and some of the original exons are now considered introns, and we cut those out. That will change the base sequence of the mRNA, which means the codons will be different, which means the amino acids will be different, which means the protein will be different. And exon shuffling is, all right, we cut out the same introns we did before, but instead of just pushing the exons all together and bonding them up, what if we rearrange them? Kind of like shuffling a deck of cards. You take out a few, and then you just put the deck back together. Or you take out the same few, and then you shuffle up the cards that are remaining. Once again, new base sequence, new codons, new amino acids, new protein. So that could help us to explain how one gene can produce more than one protein, because anytime we change the the base sequence, we are going to change the amino acid sequence. And as we said previously, and I'm going to keep saying, and you guys are going to be so tired of me saying it, but anytime you change the amino acids, you're going to change the protein. The order of the amino acids will actually dictate how the polypeptide folds up. So the order of the amino acids will dictate the folding of our protein, and how the protein folds is what dictates its function. So you're folding up a chain differently because it has different amino acids. That means that your protein is going to have a different function. So we can take the same original gene sequence in DNA, but depending on what we consider as introns and how we put the exons back together, we can actually make different proteins out of it. So just like what we were saying, intron sequences of one gene may contain the coding sequences of another. So let's say I have in my mRNA A, B, C, D, E. And originally I cut out C and D. That leaves me with A, B, E as my exons, and those made a particular protein. Well, to make another protein, maybe I'm going to cut out just B. So now I have A, C, D, E. Well, at this point, now that I have left C and D in, that means that they might help code for a different protein because we're leaving those bases in. That means we're going to have some new codons, which means we're going to have some new amino acids, which means, yes, you guessed it, we're going to have a new protein. So these are some ways that within the same mRNA sequence, within the same original gene, that we can create new proteins from the same sequence. There's also something called transsplicing, and what this involves is splicing together two different RNAs. So mRNAs from different genes in order to make a larger translatable message. So what we would have is, let's say we have a gene that's ABC over here, and then we have XYZ over here. Well, in transsplicing, we can connect those up. So we have A, B, C, X, Y, Z. Again, more bases means more codons, which means more amino acids, which means we're going to fold it differently, which means, yep, new protein. So that's another possibility of what's going on. Now, at the end of this section, I do have a video on RNA processing, one of my Science in Pajamas one. I definitely recommend you guys check that out. I actually go through and show what these would look like on my whiteboard. So it kind of makes it a little more visual. Sorry about the bird in the background. So this is kind of showing what we were talking about before. So what we're showing is, I mean, this one's more talking about frame shift on the right-hand side. But if you look here on the left-hand side, now the places where there are breaks, that's where bases were cut out. So those were the introns. So this was the original, or sorry, this was the first mRNA sequence that was processed. They're not showing us the original one. But instead of this one, maybe we have some of these other parts left in. Since we have new parts left in, new parts left in that we didn't cut out, then that means more bases, more codons, more amino acids, different protein. So like we said, only about 1.5 to 2% of our DNA actually encodes for proteins, which is kind of amazing if you really think about it. Because like we were saying, proteins do everything in our body. They build our tissues. They are antibodies and enzymes. I mean, they have an unbelievable amount of functions. They are what express the traits that make us us. And yet only 1.5 to 2% at most is what is actually coding for those essential molecules. So then what does the other 98 to 98.5% of our genome do? If it doesn't code for these important things, what do they do? And for a while, these were actually called trash DNA, which is an unfortunate name because we always thought that proteins were the end-all be-all. So if they don't make proteins, what possible function could they have? Well, we eventually figured it out because, you know, we're smart, we study, we do all this research. And what they found out is that these non-encoding sequences actually do have quite a few functions. They make different types of RNA besides mRNA. They make these things called pseudogenes. They make these things called signs and lines. Now, this is not all of it. These are just a few that we're going to mention. There's actually quite a bit of other things, but we'll talk about those later. So a good solid 1/3 of our genome codes for non-encoding RNAs, meaning that, remember we said that genes allow us to code for mRNAs. Okay, but what about the rRNAs that are located in ribosomes? What about the tRNAs that help bring the amino acids down? What about the snRNAs or snORNAs? What about all of those? So we have these many different types of RNAs, including things like ribozymes, and they have to be made by the cell. So how are they made by the cell? Well, not through the parts that encode for mRNA, because those lead to protein. So the other RNAs are actually encoded for in the other part, the non-coding portion of our genome. So a good 1/3 of our genome, give or take, is what will allow us to create these other RNAs. So see, not trash, still very much functional, just not in the same way. We also have these things called pseudogenes. Now, we're not entirely sure what is going on with pseudogenes, what they are. We just know that they are there. So pseudogenes, the prefix pseudo means fake or not really, not real, things like that. So these are essentially fake genes. And what we notice is that these pseudogenes, they might be transcribed or they might not, but they're never translated. So therefore, they're never used to make protein. We're not entirely sure the actual function of them. We're still learning. We're still doing research. We don't have all the answers. Maybe one day we will know their true function. We don't right now. That's okay. But we do know that they could be transcribed, but they will not be translated. So they're never used to make protein. And then we have these things called signs and lines. Now these actually make up a pretty large portion of our genome. And they're just these repeating sequences of bases. Now we'll talk a little bit more about them. You don't need to know too much about them. SIGN stands for short interspersed nuclear envelopes. So these are repeating sequences of DNA. They're usually somewhere between 100 and 700 base pairs in length, and they just repeat over and over. Now, they're non-coding transposable elements. That means, again, they don't code for protein, and transposable means that they can kind of move around a little bit. And then we have lines, long interspersed nuclear envelope or elements, very similar to signs, except they're much longer. So they're made up of about 7,000 base pairs, give or take, in length. And again, they just kind of repeat over and over. They are also a type of transposon. Again, that just means that they can move around. Transposons are jumping genes. And lines make up about 21.1% of our genome. So again, they make up a large part of this. Now the question that a lot of people have is, especially the pseudogenes and signs and lines, what's the point of them? If they don't code for genes, if they really don't have any function at all, what's the point? Well, some people speculate that it might be essentially a buffer zone. By having these large section of bases that do not code for proteins or even for the RNAs that help us make our proteins, that they provide a space where mutations are more likely to occur. So if you only have 100 bases and let's say 90% of those bases are help to encode for genes, and let's say mutations occur 5% of the time, then you actually have a large chance of a mutation occurring in one of those genes as opposed to in that little 10% amount of genome that doesn't code for genes. But if you have these large sections between genes where they don't necessarily have any function relating to traits or proteins or expression of traits in genes, then by expanding out the genome like that, you have these buffer zones where it's more likely a mutation might occur there than it would in the 1.5% that encodes for your DNA. So again, that's just a thought. That's a hypothesis at this point, essentially a game of numbers. The bigger the genome, the more space there is, then the more chance that a mutation can occur in one of those regions rather than in a region that actually encodes for an essential protein. So maybe one day we'll find out. As of right now, still kind of unknown, but we're working on it. All right. And then here we have other resources. Now, I would strongly recommend that as a part of this lesson, you go and view some of these because they will help. They will help you to understand the materials a little bit better. They will help you to understand the information, the different types of RNA processing, what splicing is, etc. So take a look at some of these. Now these are for the entire chapter. These are resource videos for the entire chapter. Here's my Science in Pajamas ones. You'll notice that I do have 2 of them for you guys. We have a protein synthesis one where I kind of map it out using a visual system. And it's a little long, yes, but I recommend you check that out if you're curious about seeing how protein synthesis works. I would also definitely recommend this one. It's not quite as long. It's the RNA processing. So we talk about regular RNA processing as well as show you what differential intron splicing and exon shuffling and even trans splicing would look like. So definitely recommend you check that out. We also have an Amoeba Sisters video based on protein synthesis. Not as long as mine, but because it's all animated, they do a really great job of breaking things down and showing it in a nice, smooth fashion. I do love the Amoeba Sisters. It might seem kind of silly and cartoonish, but they are very awesome at breaking down the material into understandable concepts. All right, and then we have a splicing video, which kind of, again, talks a little bit more about the spliceosome and how introns and exons are utilized to make different proteins. So might be a good thing to check out. And then there's also an RNA processing video. So again, kind of just another visual explanation. As part of this lesson, please check out at least a few of these. You can, whichever part, if you feel that you are comfortable in protein synthesis, but you're not quite sure what RNA processing is, then definitely check those out. If you think you understand RNA processing, but not quite sure what's going on with protein synthesis, check those out. I provide a wide variety of different ones so that hopefully you can find something that works and appeals to you. I'll have the link for this presentation in our comments, not comments, in the description. If you do have questions, concerns, etc., hit me up in our Google Classroom or via email. You know I'm always here for you guys. I can't answer questions and I can't help you if you don't let me know. So please let me know. Contact me if you have questions, concerns, anything like that. Come see me. Come let me know. And in the meantime, check out some of these videos and just stay healthy and stay well. All right, you guys. Take care of yourselves. Bye-bye.